Hey everybody, thanks for your time today. Like Paul said, my name is Bryan Bergman. I'm a professor here in endocrinology, and I oversee the Molecular and Cellular Analysis core. And this is kind of a unique core because it's actually composed of three separate cores, and I'm going to let those core directors tell you about their areas of expertise. So the Molecular Cellular Analysis core is composed of a lipidomic and mass spectrometry core led by Dr. Corinne Zemsky-Berry. And then we have a transcriptomic and bioinformatic core led by Dr. Kartik Shankar. And then a cellular and mitochondrial function core led by Dr. Matt Jackman. So I'm going to let the core directors take this over with their presentation. So first up was Dr. Corinne Zemsky-Berry, talking about the lipidomic and mass spectrometry core.
Great, thanks, Brian. Yeah, as Brian said, I'm the director of the Lipidomics core, and this presentation is about the capabilities that we have in our core. The Lipidomics facility contains 4 SciEx triple quadrupole linear ion trap instruments that we use for quantitative lipidomics. And it also has 2 Gcms instruments, which we use for fatty acid analysis, as well as glucose and glycerol measurements. We're located in RC1 South on the 6th floor. And these pictures here are just 4 of our LCMS instruments. So first I'll talk about the LCMS-MS platforms that we have. I thought most of you might be interested in the types of samples that we can look at, how much we would need for analysis. So this is just a little history of what we have done in our 2 years in existence. We've looked at plasma and serum, and we typically need about 50 microliters of sample for analysis. We've looked at cells, we've looked at trophoblasts, raw cells, melanocytes, and we typically find we need between 1 to 2 million cells for those analyses. And then we've looked at quite a few tissues. Muscle, placenta, brain, and lung, we need about 10 to 20 milligrams of tissue, and liver is such a high lipid containing tissue that we only need about 3 to 5 milligrams. So we take your sample and we homogenize them, and then they're liquid extracted after adding an internal standard. And so now I'll just go through the different types of assays that we have in the core. The first platform that we have, we call our non-polar platform. And this looks at diacylglycerols, ceramides, and glycosphingolipids. So ceramides, dihydroceramides, glucosyl ceramides, galactosyl ceramides, and lactosyl ceramides. This is the chromatogram that we would see upon analysis of your sample. What we like to point out here is that we can separate the 1,2 and the 1,3 DAGs, and why that's important is because in most cases, the 1,2 DAGs are the biologically active DAG isomers, and so we can separate them and give you information on the 1,2 versus 1,3 DAG isomers, as well as all the other lipids listed in this slide here. Our second platform we call the polar platform. This looks at sphingolipids as well as acylcarnitines. So we look at sphingomyelin, sphingosine, and acylcarnitines. For our acylcarnitine analysis, we go between 5 and 18 carbon chain lengths, and that's shown in this chromatogram here. And then for sphingomyelins, they're very similar in mass as well as mass spectrometric characteristics. So we can separate the sphingomyelins from the PC chromatographically, and so we know that we're looking at sphingomyelins in your sample. The third platform that we have is a phospholipid lysophospholipid platform. This is a normal phase-based separation technique where we're separating the lipid classes from each other. And so for this, we can get all of the phospholipids listed here. We can do diacetyls, we can do ether, as well as lysophospholipids. And then we also look at triacylglycerols. This uses a reverse phase method where we separate by lipophilicity. So it depends on the number of carbon atoms as well as the degree of saturation of the tag molecule. And then finally, we have a nicotinamide assay. These are just biologically active lipid mediators derived from arachidonic acid or linoleic acid. Their HODs, heats, leukotrienes, prostaglandins, as well as thromboxanes are in this assay. And then we also, so those were our LC-MS assays. We also have GCMS assays. So we can do free fatty acid analysis for these analyses. We typically use about 25 microliters of plasma or serum, and for cells, about 1 million cells. We can do content and composition as well as isotopic enrichment. And then for our glucose glycerol assays on the GCMS, the plasma serum amounts are about 100 microliters, and we can do content as well as isotope enrichment here. So this is the information to get in touch with us. The website is listed there. We list out all of our assays that we have that are available to you that I spoke about today. You can contact me for more information, for quotes, for some advice on samples for submission. And we do have 2 pricing tiers. The first is that the Lipidomics staff kind of takes care of all of the sample prep. So that's a more expensive option, but then there's a less expensive option where someone from your lab can come in and do the extractions. And that is all that I have.
Super, thank you. All right, Kartik, you want to take it over with transcriptomics and bioinformatics?
Okay, so thanks so much. I'm going to tell you a little bit more about the transcriptomics bioinformatics core within the NORC. And this is probably in its kind of new incarnation since I have been here for a couple of years, a revised version of the kind of pre-existing core within the NORC. So I'm going to tell you what we do, how you can access our services, and what we are kind of planning to expand on. So the goals of this core really are to provide a one-stop sample to publication pipeline for primarily bulk RNA sequencing, but we also do microbial ecology analysis and then bioinformatics for epigenomic analysis. And I'll walk you through what this really means and what kind of applications you can look at in the context of your own work. We also provide secondary tools for data interpretation and visualization if you already have existing raw data that you have generated in the lab or through another core. And then really the overall goal for the core is to promote collaborations and also be kind of a point of development for new investigators who have pilot projects, many times coming through the Colorado NORC itself. So this is kind of a broad schematic of the pipelines that we have in the lab. I'm going to focus on the top one here, which includes bulk RNA sequencing. And I'll walk you through kind of the modular approach we have. But the idea really here is to examine gene expression within tissues. We do this both within human tissues or mouse cells or tissues or in vitro cultures. You can come in with either tissues itself or prepared RNA, and then we can make libraries. We partner with the CU Genomics Core for sequencing services, and then we have our in-house pipeline for data analysis. The same kind of walkthrough can be done for chromatin and DNA methylation type analyses, where you can come in with DNA or a tissue, and we can isolate chromatin, perform CHIP again, and then we have bioinformatic pipelines for ChIP-seq and DNA methylation. And finally, we have a very robust pipeline for doing microbial ecology analyses, and these typically are done in feces of either human origin or mouse or animal stool, or can be contents from the gut. And really require isolation of metagenomic DNA and then preparation of either amplicons of particular microbes or the entire metagenome sequence. And then we have application-specific pipelines for the microbial ecology analysis. So, so there's a wide variety of kind of applications that we can do. The resources of the core primarily are wet lab space in the RC2 building. On the 5th floor, we have equipment to do all the molecular methods, including sample preps in modular formats, so they can be done in 96-well formats, and the equipment to prepare DNA sequencing libraries. And then we really take over at the step where we get raw reads from the genomics core for bulk RNA sequencing. And so for those tools, we have two major kind of hubs. We have a Linux server, which has 80 threads and has all the kind of up-to-date software on it for both bulk RNA sequencing, ChIP-seq, and a variety of microbial ecology workflows. And then we have a separate data storage server, which can be used for archiving data. So here's kind of the broad overview for what we can do. We can accomplish sample prep tasks, so isolate either DNA or RNA from tissues and cells, or we can isolate DNA from bacteria, typically from feces, which is the initial point of kind of starting a project. And then if you already have these nucleic acids isolated, we can prepare DNA libraries or amplicon libraries for microbial sequencing. And then we can do tasks related to the data analyses. We do have the workflows to support single cell RNA sequencing data analysis that are post data generation. And so we have users enter the 10x genomics data analysis pipeline after they've generated data with the core. So the workflow is modular so that you can essentially arrive to the core with any point. You can either arrive with the sample itself, or you can come with data that has already been generated from the genomics core or even within a lab. And then we can help with aspects of both primary data analysis, which typically includes cleanup, reads, assignment, alignment, and then or secondary data analysis that typically includes RNA sequencing quantitation, such as identifying differentially expressed genes between groups. And then there's tertiary data analysis that's typically interpretive analysis like IPA and Go term enrichment and things like that. And this is just a kind of an example data analysis pipeline for bulk RNA sequencing, but similar kind of stepwise analyses exist for microbial ecology and things like that. We also have two RStudio servers in the lab that have a lot of R packages that can be then utilized for typically the second and the third steps of this process. And like I mentioned, we partner with this genomics core for sequencing. We don't do any sequencing within this core. How do you get started? Depends on the kind of specifications of your project. We encourage you to get in touch with myself or with the core lab personnel. The cores really run both by myself and then by Pooji Jambo, who's a senior PRA within the Department of Pediatrics in the section of nutrition. And I know this is kind of very 30,000 feet, but I'm happy to answer questions. Yeah.
Excellent. Thank you, Kartik. All right, next up for the Molecular and Cellular Analysis core is Dr. Matt Jackman, talking about cellular and mitochondrial function analysis. Matt, take it away.
Okay, so my presentation's a little bit different. First of all, everybody else that has talked so far today is talking about services. I don't have any services. I have equipment, and so it's a shared equipment lab space. And so we'll go through most of the equipment that I have, and then I'll tell, basically, we're going to try and teach people through the course of this endeavor. And so, next slide. Where's my slides? Too many monitors, sorry. Okay, so these are the resources we have available. They're not in any particular order of use. It's basically how I grew up using the instruments. Been doing this mitochondrial stuff for about 25, 30 years. Back then, they only had clerical electrodes, and so that's what we used. I'm lucky to have two now, but we don't really use them too much anymore. I don't think Brian used them maybe 5 years ago. We then upgraded to the Ouroboros O2K respirometers, and they are basically a fancy Clark electrode that's hopped up on steroids. They're a lot more sensitive, they're a lot more controlled, and they're a lot more coupled with everything. We then, about 3 years ago, Jed Freeman was running the XSB96 Seahorse analyzer. He left for Oklahoma. It landed up on our lap, and that's when we started the core. And then we also have the Biotech Citation 1 cell imaging system. And so this system is a standalone system. We also have people that use it for imaging, but it's also coupled to the Seahorse analyzer for normalization procedures. I don't think any of these, every one of these pieces of equipment have pros and cons. Not one is any better than the other. They're complementary. So if you can't get something done on one system, you can move to the second system and can hopefully get it done. So then I'm going to just go through and start talking about, this is the bread and butter. The Seahorse gets the vast majority of all the use in the core. It is a high throughput system. It's got 96 wells, so you can get a lot of stuff done in a short period of time. It gives you glycolysis and mitochondrial oxygen consumption. It is somewhat limited in that it measures things, number one, at normoxia. It measures basal respiration rates, and then you can get maximal uncoupled respiration rates. We can do other things in the other instruments that would replicate those as well as some other things. The adjunct reps are actually going to be on campus this afternoon, and one of the questions I'm going to ask them is, what is the O2 content and when can I see the O2 content? Right now, all I know is it's the O2 rate, so it's the rate of oxygen consumption. I don't really know how low the O2 goes or how you would know if it goes too low unless you started seeing inhibition on your cells. Training on this is the easiest one because I don't really have to do anything. The adjunct has lots and lots of videos. They have seminars on use, data analysis. The field representatives have been excellent. They're very responsive. They get back to you pretty quickly. After you look at the videos, you might want to shadow another user. And basically by shadow, you're just tagging along, watching them do what they're doing and asking questions as you go. If you're not comfortable with running it on your own, just let me know and I'll run your first assay with you. Not a problem. There's only one hard and fast rule about the Seahorse, and that is that you cannot leave the top on the plate when you put it in the Seahorse. That's catastrophic, and we will be losing the machine for months at a time. It has to go back for repair. In the picture, you can see the citation next to it, and that is for the normalization again. Basically, what you do on Seahorse in one of your last injections, you put in a hoax stain. It's basically the same as DAPI, only it's more permeable to the cells. It goes into cells, takes about 30 minutes, so you can add it with your last additions of inhibitors. It's not going to kill the cells. That's a humorous story on its own. You take it out of the Seahorse, you put it in the Biotech, in about 20 minutes, it has taken about 4 gigs of data, and then it converts it to about 96 numerical values for each one of your wells. Generally speaking, you put the hoax stain in last, but we actually had one user that put it in first. And our initial interpretations were that the data was going to be bad. It does not kill the stain. It does not kill the cells at all over the course of the experiments. So that's not a problem. This might be the only tandem Seahorse Biotech combination on campus. And right now we're getting quite a few calls from other Seahorse users that want to use our system for normalization because they don't have the setup. And it's not a bad idea because the citation is pretty costly and then the software costs far more than it should for this counting procedure. The second most used instrument is the Ouroboros O2K. We have 2 of them. which means we have 4 chambers. 4 chambers versus 96 chambers, it's a big difference if you're looking for throughput. One of the O2Ks is a little fancier than the other one. You can do reactive oxygen species, you can do membrane potential, you can do a lot of other perturbations. It's got a little pump on there where it can pump in any drug you want at 0.1 microliters an hour or minute. But the system is very slow, it's very sensitive, but there's a lot of benefits to it. The Seahorse has 4 ports. The Ouroboros has 1 port at the top. You can add in 1,000 things if you want over the course of time. You can measure respiration at any O2 that you want. You can take it to hyperoxia, you can take it to hypoxia. If you start to run out of oxygen, all you got to do is open the top, let it breathe for a little bit. O2 diffuses from the atmosphere down into the well, and you're back up to basal levels, and you just start your experiment all over again. So you can literally do 6-8 hours experiments with this thing as long as your tissues stay alive. So you have a lot more flexibility, but going with that flexibility it is pretty labor intensive, and it takes a lot of training. We're not just going to let you go on this one on your own. There's not a whole lot of videos on it. We have to sit with you for quite a while and then coach you through it. Most of the samples that we're using in here are permeabilized muscle fibers. Every single fiber that you put into the chamber acts as their own control. So unlike the Seahorse, where they blow in 1 concentration of an uncoupler or 1 concentration of the substrate, you can sit here and you can titrate it in over the course of time. If you go too high, cells will die, and you do not want to kill your human muscle tissue. If you do go too low, you're not going to get maximal rates. So in the end, once you get comfortable with the system, there's a lot more art that goes in with the science. You just kind of know when you can add stuff and you know when you can't add stuff. The other thing on the system is we no longer use oligomycin. Oligomycin is used all the time in the Seahorse, and you get to use it over there because you throw the cartridge away. If you put it into this system, it's a bear to get out of it. We have colleagues in North Carolina that they will not use the system for another 7 days at 100% ethanol. We've lost a lot of data because people were not cleaning the instrument as well as they should have. And you don't know it until the end of that 6-hour period. That's the kicker. Everything looks like it's going well. You get to the end and you're like, oh my God, this was horrible. So in closing, again, no one instrument that we have is going to be perfect for what you need. Whatever instrument you need is going to be defined by the question that you have. And so if you have any questions, you can contact me here. That's the lab number. I'd say that 90% of the core users know my cell phone number by now, so you'll get that sooner or later if you want it. Thanks.
All right, so I think we've come to the end of the hour, and we've taken the full hour on you. So if you have any questions, I think the best thing to do is to reach out to the core directors and of the directors of each of the components of the individual cores to get more information. So thanks for joining us, and I hope this was informative. Thanks for being here.